And welcome to the Evidence Reasons Academy. Let me make sure I turn on the correct mic. And I'm joined by my co-host, Cindy Lincoln, today. We're going to study molecular biology. This is kind of our sixth installment, part 6. And we're just kind of, this isn't a structured program. I'm kind of learning how to maybe communicate this to people who aren't full-up biology students, and trying to get it, trying to see how quickly this can be taught and effectively, and try not to make it too painful a process. And the idea is to equip people in this field. Now, molecular biology is unfortunately a little bit of a misnomer because, strictly speaking, they talk about they talk about DNA transcription and then RNA translation. That's a lot of molecular biology, but the way it's defined now and the way it's taught. But really, if we're talking about the molecules of life, the molecular basis of life, the proper discipline would be considered biochemistry. In fact, I have a book, McKee by McKee and McKee, they're a husband and wife co-authors, that says biochemistry, the molecular basis of life. So in a sense, the true molecular biology I consider to be biochemistry, but just for historical reasons, the discipline of molecular biology ended up focusing on DNA transcription to RNA and then RNA's translation to proteins. That's just how, historically the terms quote unquote evolved. And so we will focus on basically what is considered traditional molecular biology. And we're going to do that also from an intelligent design perspective, just to see that this does point to an intelligent designer if we study it in a certain way. And that's also one way of making it interesting. The way that we're going to try to do this is we're going to actually watch a low quality version, meaning it's low resolution, of Illustra Media's Unlocking the Mystery of Life. They have generously allowed the material, although it is copyrighted, they have allowed others to use it on YouTube. I'm not gonna, I get no monetary benefit from this on this channel. This channel is not monetized right now, and I'm very grateful for that. We are appearing via recording because that enables us to use copyrighted material where the copyright owner has given permission to show it on YouTube. So they may give permission, but then YouTube has a restriction that if you use it in a live stream, you could get some actions taken against you, like a copyright strike or whatever, because live streams are also enabled for gathering money. I don't know the reasoning. They allow it for recording, provided the copyright owner gives permission. But even if the copyright owner gives permission to use their material, if it's a live stream, YouTube will just shut that down. So that's just the way it is. So Cindy, do you have any remarks?
Hi.
Hi.
No, I can't wait to see it.
All right, maybe we'll just get to it. Let's just say a quick prayer. I hope you enjoyed the opening.
I did very much.
Yeah. Let's just say a quick prayer. Lord, please be with our viewers who are joining us via recording. Thank you for the work of Illustra Media, and just guide our conversation as we reflect on what we're watching. In Jesus' name, amen. So I'm going to now bring up the first video. So let's see. Unlocking the Mystery of Life, chapter 1 of 12. Okay, here we go.
In 1993, Professor Philip Johnson of the University of California at Berkeley invited a group of scientists and philosophers to a small beach town on the central coast of California. They came from major academic centers, including Cambridge, Munich, and the University of Chicago, to question an idea that had dominated science for 150 years.
I think Pajaro Dunes represented a turning point for many of us. Individually, we all had questions about evolutionary theory, but when we came together, each person brought something of their own to the table. And suddenly we all had a glimpse of a new way of looking at life that none of us had individually seen before.
I would have to say that this was an intense period of time in my life. It just seemed that there was something here much more intellectually satisfying than the view that I had held up until this time.
Looking back on it now, I think that gave me the motivation to actually look at the evidence and just see where I thought it pointed.
I realized that this was bigger than any one person or discipline, and this was the beginning of a community of scientists who are now willing to face the fundamental mystery of life's origin.
I sometimes wonder why anybody talks about anything else, because this is the most interesting topic there is. Where did we come from? How did we get here? What brought us into existence? What is our relationship to reality as a whole?
You look at the incredible diversity and complexity of life, and inevitably the question arises, what brought all this into existence? Was it simply chance and necessity, undirected natural forces? Or is there something else going on? Is there a purpose, a plan, a design due to an intelligent cause? I think that is the fundamental question.
The scientists who came to Pajaro Dunes set out to re-examine the mystery of life's origin. For each had significant doubts about widely held evolutionary ideas. Among them, biochemist Michael Behe questioned how natural processes could have assembled the intricate structures found within living cells. Dean Kenyon was an evolutionary biologist who no longer thought that chemistry alone could account for the origin of life on Earth. And Stephen Meyer, Paul Nelson, and William Dembski were seeking a new approach, one that could explain the origin of the genetic information encoded in living organisms. These scientists and philosophers began to formulate an alternative to the central theory of modern biology, a theory born in the mind of a British naturalist. His name was Charles Darwin.
Cindi, are you there?
I am, but I lost the hookup we had before. Are you hearing that bad noise from my laptop?
It's tolerable.
Good.
So we'll just go with what we got. I'm going to mute myself. Do you have any comments before I queue up the second part of 12?
No, I think we saw that last week, that first part.
And that's fine. We'll just play it again.
Actors.
Yeah. Actually, those are the real people.
Well, I mean that, yeah, the players.
Yeah.
Go for it.
So they're the older generation that eventually, a lot of them ended up in the Discovery Institute. They are now senior fellows. The next generation of fellows are like Joe DeWeese and Casey Luskin. And you were part of that meeting that one day.
Now, so how old is this film? How old
is-
were they back? I mean, how many years ago was it when they were at that conference?
Probably 22 years, maybe even 30 years ago.
Wow.
But it's amazing. This is a lot of what they say is prophetic. This is why it's also powerful. And when I was thinking about what the videos we should watch, this one still kept coming up. I said, it's a classic. But I'd like you to, one of the characters there is Dean Kenyon. He was an origin of life researcher, and he switched. He became a creationist. So he's looking for a naturalistic solution, then became a creationist. Now, I don't know if he was a Christian before. There were a lot of Christians that were looking for naturalistic solutions, and that's fine. You know, they may have had a different interpretation of Genesis. So I'm not saying he necessarily was not a Christian when he was an origin of life researcher, but he was actually one of the premier origin of life researchers at the time. He then believed in intelligent design, and he got in huge trouble. So just kind of file away when you see Dean Kenyon, just especially.
You sent me a link for maybe his testimony. You sent me an email. I
Yeah.
had asked about it.
I'm sorry.
I had asked about him, I believe. And
yes, it was that one that I sent. That's Dean Kenyon. And they're probably better articles than the one I sent you, but I just wanted to get you started. So let me get episode 2 of 12 now going.
In 1831, Darwin, then 22 years old, set sail on a 5-year survey expedition for the British Empire. He journeyed from England on the HMS Beagle, traveling around the southern tip of South America, then north toward a chain of volcanic islands in the Pacific called the Galapagos. On this desolate archipelago, 600 miles off the western coast of Ecuador, Charles Darwin encountered an extraordinary array of birds, reptiles, and mammals, the likes of which he had never seen before. For more than a month, Darwin studied plant and animal life, took extensive notes, and collected specimens. Then he left, never to return. 25 years passed as he developed a theory about how the diverse forms of life on Earth had originated. In 1859, Darwin published a book titled On the Origin of Species. Its impact on science and ultimately all of Western culture was dramatic. Darwin argued that all life was the product of purely undirected natural forces, time, chance, and a process he called natural selection.
For 2,500 years before Darwin, most prominent scientists and philosophers, people such as Plato or Newton or Kepler, viewed the world as the product of some kind of design or plan. But a fundamental shift occurs with Darwin's idea of natural selection, and a real change in scientific philosophy is set in motion.
Darwin was not the first scientist to propose a theory of evolution, but he was the first to offer a plausible naturalistic mechanism that could produce biological change over long periods of time. To understand how natural selection works, consider the finch populations Darwin encountered on the Galapagos Islands.
13 species of finches inhabit the Galapagos Islands, and they vary subtly in terms of their body size and shape of the beak. Darwin returned to England with 9 different species of these birds.
According to contemporary Darwinian theory, differences in the sizes and shapes of the birds' beaks are the direct result of natural selection. One example often cited involves species of seed-eating finches. Following seasons of heavy rain, small, soft seeds are plentiful throughout the islands. Birds with short beaks can easily gather food. However, during periods of drought, the only seeds available are encased in hard, tough shells that remain on the ground from the previous year. In these circumstances, only birds with longer, sharper beaks can crack the shells and eat the seeds.
Those birds with the longer beaks survive because they can reach the food source, whereas other birds cannot. That long beak then confers what biologists now call a functional advantage. The finches with smaller beaks, unfortunately, die out from starvation because they cannot reach that food source. If the drought conditions continue, the environment causes a change in the features of the finch population as a whole. Over time, the long beaks are passed on to succeeding generations because those beaks enable the birds to survive.
Natural selection was a powerful idea. Physical variations that proved advantageous would be inherited by succeeding generations. Through this process, populations would be altered, and over time, fundamentally different organisms would arise without any form of intelligent guidance.
Darwin wanted to explain everything in the history of life in terms of undesigned, unintelligent natural processes. And when he looked for an explanation, what he found was that a process he could observe in domestic populations also operates in the wild. Now, Darwin himself was very familiar with domestic breeding. He himself studied pigeon breeding. And he knew that for centuries, human breeders had been able to make dramatic changes in populations by selecting only certain individuals to breed. Darwin merely suggested that this same process operates in the wild.
For Charles Darwin, natural selection explained the appearance of design without a designer. There was no longer any need to invoke an intelligent cause for the complexity of life. In effect, natural selection became a kind of designer substitute.
Today, Darwinism is generally assumed throughout science and the academic world. Yet, despite its wide acceptance, a growing number of scientists and scholars, including those who met at Pajaro Dunes, now challenge key aspects of Darwinian theory.
When we came together at Pajaro Dunes, we certainly didn't agree on everything, but we did share a real dissatisfaction with the mechanism of natural selection and the role that it was playing in biological explanation. Natural selection is a real process, and it works well for explaining certain limited kinds of variation, small-scale change. We have lots of examples of that, in fact. Where it doesn't work well is explaining what Darwin thought it could, namely the real complexity of life. We have the finch beak, and then you've got the finch itself. A minor change in the structure of the beak versus the origin of the organism itself. These are different scales of phenomena. These are different kinds of problems. And the important problem for biology is to understand where natural selection works and where it doesn't, and why there's a difference. Evidence is very powerful, and all of us had the sense that if we let that evidence speak for itself, that it would lead us in a very different direction, away from natural selection and towards a different conclusion about the origin and nature of life on Earth.
You have any thoughts, Cindy?
I'm still confused because evolutionists will say the opposite of what they just said. They just said that Darwin's book proposed that random processes, and now they will tell you, oh, well, it's not random, because it's real obvious when you look in nature. You see symmetry, you see pattern, you don't see random. And
Well, I'm sorry to stop you. They said natural processes, time, and something called natural selection. They're
I heard that too.
component.
Right.
Yeah. So if you say Darwin proposed this plus natural selection, then you're good. So it's not just pure random chance, it's also natural selection.
And I think that's what they have said when I've objected with them, is that natural selection isn't random. It's choosing beneficial things.
Okay, there is a problem with that. One, the outcome may not be random, but it doesn't lead to complexity. We know experimentally now. At the time of this video, we didn't have as much evidence as we have now. So what reproduces more is, when they say natural selection, it means survival of the thing that reproduces the most.
Or the thing that doesn't die off.
Right. Now, do you remember my Christmas present for you? It talked about how natural selection. Okay, this would be an opportune time to show how natural selection might really work. And it's going to star someone that I think he really had it coming to him. I filmed this. We were in a discussion, and he said the stupidest thing, and he really meant it. And I said, dude, I'm going to hammer you for that. I'm not going to let you go, because you made my case for me, because he demonstrated what happens in real world natural selections. So if you give me a moment, I'm going to cue this up for us. And this was my Christmas present to you and Rebecca. And this is the guy that was pleading. He was pleading. He said, please come on my channel. You know, Sal's misleading you. And I just, like, I took exception to that. I said, I'm going to take care of this. I'm going to take care of this bum. And I'm also going to give a nice Christmas present to
Pesky
Cindi.
fly.
So let me find it. I hope I put it here. Here it is. Christmas. I'm going to hang on. Let me bring this up. So this is how natural selection works. Even by his own admission, this is how it works. So let me bring this up. Share screen. And I want to share a Chrome tab. Early Christmas gift. Here you go. All righty. Let's start it.
It could well be the best thing for the human race to get dumber and dumber until we lose our minds entirely. It could well be the best thing for the human race to get dumber and dumber until we lose our minds entirely. It could well be the best thing for the human race to get dumber and dumber until we lose our minds entirely. I mean, that may end up being the most reproductive, successful thing, right? But so be it.
Oh, Lord.
Okay, there you have it. All right. Now, aren't you glad you turned him down? What a waste of time. And I said, you know, when you told me that, I said, I got to let you see him make the case for me. He's actually, I was saying, dude, natural selection can result in loss of complexity. Isn't that right? So that meant you could be losing intelligence and sicker. He said, yeah, it could well be we're getting dumber and dumber until we all lose our minds. It's like, yeah, that's natural selection for you. So the stupider might be the most reproductively successful. That's where it's going to end. And I have since got all these papers that show, like, for example, it's kind of sad, honestly, very intelligent women, 25% of high IQ women, they'd be classed in the genius range, have 25% have no children. They there are various reasons that women with elite minds like that just do not have kids. In natural selection.
No comment.
No, well, I'll tell you how. I'll tell you. Well, you know, I mean, in the case, I have a cousin. She's brilliant. She got her PhD at MIT, and she was at a party one day, and the guys were introducing themselves, and they started saying, well, tell us about yourself. She said, oh, I just graduated, and they said, oh, did you get your bachelor's? She said, oh, no, I just got my PhD, because she looks so young. Oh my goodness, I think she may have been a really young PhD, like at age 24 or something. Just, she's bright. So she looked like an undergrad who just graduated. They said, did you get your? She said, no, I got my PhD. And they said, oh, where? She said, MIT. The guys just went, okay. She's since found a wonderful guy since those days, but she was relating what it was like to meet guys. She would just intimidate so many of them. She met Mr. Right, and they're happily married and have 2 grown kids. That's my cousin Libby. But it goes to show that the individuals that reproduce more of their own lineage isn't necessarily the one that's going to be more complex or have more versatility and capability. And experiments with bacteria are really bearing that out. So you keep hearing about Lenski's experiment, and it's a disaster. They said, look at all the new functionality, and I just laugh. Why don't you read the fine print, guys? The same author said, look at all the versatility that was lost. The genome decays despite sustained fitness gains. I said, this is a disaster for your theory. You're losing genes? And you think this is a good thing? You're supposed to be building and accumulating, not losing versatility and capability. So yeah, if you want to say it's not random, it's like, well, look what it actually does when we actually study in detail, now that we know molecular biology. Because we could actually see DNA being lost. We didn't have that in Darwin's time, but now we could actually see pieces of the blueprints being lost. And this is the difference between 30 years ago. Now we can just hammer them. If someone wants to debate me on this, I'm going to hammer them. I'm just going to say, look, I got secular peer-reviewed papers. I don't have to reference creationists. And then I'm going to show that video of Mike. If I debated him, I'd say, Mike, you made the point for me. How about I replay what you told me when we were having this argument about natural selection? I'm not going to let him live it down.
So that was the whole context? He was saying natural selection was not random?
I was pointing out to him, natural selection can destroy, right? And he said, yeah, that's obvious. I'm just like, okay. And so he admitted it.
See, this is a new concept for me. I've only heard natural selection is a good thing. I've never heard that it can destroy.
All right. So now aren't you glad you're hanging out with me? I'm teaching you right. Because it's not that I'm smarter or anything else. It's just that so much has happened in the last 5 years alone. You just have to be willing to suffer through some reading of the literature, and you'll see this.
Well, and the point still remains that it's still, well, I don't know. If it's not random, it's intelligent. If it knows, hey, I have to select this one and not this one, that's intelligent.
It's foresight. And you see, this is a word, this is kind of dishonest rhetoric. At this point, it's dishonest. Maybe it was just naive in Darwin's time. What he described is fantasized. It's a fantasized mechanism. It's not natural, and it's not really selection in the sense of like an intelligent, goal-driven
person selecting anything.
Right. Because selection often means you have foresight. So when you have a goal in mind,
And you're thinking.
right. So like if you're trying to build an aircraft, you're choosing parts and designing the parts with a goal in mind. The thing about
having the parts there is a big part of it. You can't select what's not there.
Oh, exactly. So brilliant. And they'll say, well, it selects something that's kind of almost there. And it's like, no, you haven't proven that. Because what we're finding is if you have something that's half broken, that gene is sometimes probably gone for good. Unless there's an infusion from an external source, what they call sometimes horizontal gene transfer, but we don't have to go there. But
well, and they are trying to separate evolution from origin and saying, that you can extrapolate from different beaks to different kinds. And the whole question is still, in my mind, it's not what goes on in the planet naturally. It's, was there a God needed to create us or not? And if you're going from nothing to something, you need a lot more than selection.
well, that's for the origin of life. But once you have already an organism that's existing, for example, all these animals called metazoans, these are animals that have embryos. There's some animals that don't have an embryo phase. I don't know all the details, but evolutionists themselves have said we call them metazoans. All the metazoans, not all of them, but this protein called collagen is something that's only in metazoans. And collagen is really hard to evolve. It would resist an explanation by natural selection. And if anyone wants to argue with me, go have at it. Because I'm not going to make you guys. I'm not going to make it an easy argument for you. If one studies it, because we actually now know the molecular details of collagen, and it's not a simple protein. For a collagen to come about, you need 5 other proteins to already exist, and even if they do exist, they have to be modified to be able to work with a collagen. And each of those proteins is complex, meaning if a few key parts are knocked out, it's gone. It's not quite as severe, but comparable to a password. If you don't get all the letters right, it's gone. So in the case of some of these complex proteins, there might be 10% that really have to be there, and that would be like 100 amino acids. So this is like having a password that's 100 amino acids, and you got to get it right. Otherwise, it's just compromised. Forget it, And the difference now is we can really argue at the molecular level and really take it to them. Before it was kind of vague. We kind of knew we were right, but now we have a lot more evidence and understanding of biology. So just like what I gave to you today, you kept hearing natural selection builds. Now you learned something. Natural selection can destroy. And in fact, Michael Behe published in secular peer review. All he did is he reviewed all the experimental evidence where we actually were very, very careful to monitor what happened. He said the overwhelming majority of things that are selected for are for loss of function, not gain. And certainly not.
What?
Yeah. Hey, and if you doubt me, if you're on a debate stage, use the Christmas gift I gave you. Do you want me to play it for you again?
No. No. Because, I was always taught it would select something better. You wouldn't think
Okay, let me give you a good example. Just think about this. You have two individuals. One is fast and one is smart. They're both traits you would like to have, right? I mean, you know, one individual is smart, the other one's fast. Now a lion comes along and he wants to eat. And he's chasing these two individuals. One is fast and one is smart. What happens to the smart guy who can't run as fast? So now you've just dumbed down the population because you had one guy with a trait that's fast and the other guy trait that is smart. And you see, we call that interference selection. And someone will say, well, if there's one individual both smart and fast, then he's okay. It's like, okay, I grant you that. But let's say you're at that stage where, you know, one guy emerged that was fast and one that was smart. So don't assume even in that scenario it's going to work. And there's more subtlety to this. What if you have a population where every kid has at least one defect that the previous generation didn't have? One defect. Turns out there's 100 defects, subtle, at the DNA level, on average per individual per generation. So each generation adds 100 more to your genome. That's well established. What ends up happening is natural selection. It will be the survival of the fittest, but the fittest genome was the parent. It's the fittest among the defective siblings. And when you actually think through it, it's like, how are you going to build complexity? So let's take an extreme example. The parent passes off, generates 10 birth defects in each of the children, or at least on average. So you might have one with 9, one with 10, a third kid with 11, something like that. So even if you take the best, which is the kid with only 9 defects, you still degraded the genome. And that's what they call the problem of mutational load. And when people started to think about that, it's like, oh, this is problematic. Why isn't this in evolutionary textbooks? You don't, this is so brutally obvious.
I heard somebody arguing that against genetic entropy, that it wasn't, you just said that it was well established. Why would he be saying
who is arguing that?
I can't remember who it was.
You know what I do in that case? Do you know what I do in that case? I say, can you cite, okay, let's not talk about viruses. Let's talk about the human genome, because the genealogy of Christ deals with humanity. And for a lot of Christians, we're focused on that. Tell me one major geneticist that thinks our genome's improving. Name one. Because I could easily name 5. There are probably at least 10 who are top tier. I don't know a single top tier that says our genome is improving.
Well, for me personally, I had a section on that in the Science Center. I didn't call it genetic entropy, but just as a teacher at the age of 60, I can go back and see how kids used to be and how kids are today. And it's not an improvement. It's a decline.
It's a decline.
And I know personally as a mom and as a child, how many things do parents teach their children that the children reject? How much wisdom is lost with every generation?
Well, but we're talking about genetic loss, not just cultural decline.
Well, I'm talking wisdom.
Yeah, but I'm talking
ability to do things, ability to memorize, ability to.
Yeah, we're losing ability. Hey, and I could play you that clip again.
Mm-hmm.
That's a diehard atheist evolutionist saying, Cindi, please come on my channel. Please come on my channel. And I was just laughing. I said, don't go on his channel.
Well, and it's still, you've still got the fundamental nature of selection requires something to choose from. And if you start from the whole molecules to man thing, molecules don't have any more advanced organisms to choose from.
And exactly. And so just modify that a little more. What if what you have to choose from is worse than the prior generation? Where they're all defective. They're all slight defects in every human being that the parents didn't have. We know that.
Because of the nature of copying. Copying
Yes.
always has a slight mistake.
That's exactly why molecular biology, you begin to see it. We didn't have that in Darwin's time, but now it's so easy to see, and you can't run away from it. We can't run away from it. We thought natural selection would clean it out. It would if we weren't having so many mutations, but when we have so many mutations that every kid has at least one, and it turns out to be more closer to 100, there's no way to reverse this. And I've been arguing with the guys. It's like, guys, there's not any way around this. And I said, you evolutionists out there, if you really want to challenge me out there, find me one major geneticist that thinks we're naturally evolving better. In fact, it's getting so bad, they're suggesting 1, eugenics, 2, genetic engineering. And genetic engineering is intelligent design. Isn't that funny that to rescue the human race, you have to invoke, you evolutionists are now invoking intelligent design to rescue the human race. Doesn't it dawn upon you that maybe that's how we came about? If you're needing it now to be fixed, maybe the thing that created us, we need to appeal to him, beg for mercy, and maybe he can heal our bodies. I mean,
Take us home. And okay, even if we give it to them, let's say the humans are getting better. Let's just pretend.
Okay.
And then extrapolate that out to molecules or monkeys and say they're getting better. Are they getting different? Are monkeys getting so much different that they're actually becoming human-like? You see where I'm going with that?
No, but I would encourage you
The nature of evolution is to change from one thing to another. There's not a selection for that.
Well, I would encourage you to study the genetic entropy argument for me a little more. And if you think I'm giving you the deal, that's why I gave you that Christmas present. You heard it from the monkey's mouth himself.
I agree with genetic entropy. I can see it. And we studied about how the DNA transcribes, and basically like a copy machine, it copies itself, and copy machines have slight degradation every copy. That's not hard to understand. I'm just saying, even if it wasn't so, there's still enough to select from when you're a molecule or when you're a monkey. There's nothing to select from in your gene pool that will make you into a man or a more complex organism.
Well, let me just say, you'll be able to articulate that argument the more that you keep studying. It'll become really, really clear. You'll have really strong arguments. So for nothing else, one good takeaway you have from here is not what happens in nature is not accumulation of complexity. We now know it. We know it theoretically. We know it experimentally. And if there's any doubt as far as humans, you ask your opponent, cite me one geneticist that thinks we are making brand new genes and we're improving versus decaying.
Well, and even if you look at the finches, And you're saying, okay, natural selection selected for a certain type of finch. How was that? What did you just call it? Accumulating complexity?
Accumulating complexity. Well,
Even if finches weren't accumulating complexity, they were dying off the less.
well, there are limits. There are limits. There may have been some that are advantaged in that environment, but what happens is you lose versatility. So what it means to lose versatility is in one environment, maybe intelligence is favored, but in another, it's not. It's some other trait. And so what we find is when you have strong environmental pressure for one trait, you end up losing other good traits.
Well, yeah, I mean, that's what they said. The finches that don't have the stronger beaks die. So then you lose the
Right.
finches that are able to go, eat something different.
There could be, we're finding out there's often trade-offs when you do that. So,
So how is losing finches accumulating complexity is the point I'm trying to make.
right. But the more molecular examples you get under your belt, you'll actually start to see, it'll start to be really, it'll be more powerful. You'll have examples. And so that's why, rather than kind of making it up in your own, kind of from your own observations, when you can get to the point you could cite professional literature that's written by evolutionary biologists, it's going to be devastating. And there are ways to frame this where you don't have to read all their technical, but you know the paper well enough and you know the meaning of it. So, for example, okay, let's see. This is really easy. Despite sustained fitness. Okay, I'm just going to show you a title here. This is 2017. And we'll do some more molecular biology. I'd like to try to finish the Illustra media. Look, see this? Genomes decay despite sustained fitness gains. That means fitness gains means, the thing that's reproducing the most is reproducing the most, and it's going to be the most representative. So this is analogous to that finch that has the stronger beaks. But so they're showing that these things have sustained fitness gains, but the genome is decaying. They're losing genes. They're losing.
What do they call it when bacteria resistance? Is that so? It may be becoming resistant to something, but
a lot of antibiotic resistance is loss of function, not all of it. Now, the way that some bacteria acquire new function is it's called horizontal gene transfer. They may get it from other bacteria. They may find the parts that someone else has and then incorporate it into their genome. But if they don't have that access, the genes are gone forever. This is known, it is problematic. If that gene is lost in the population, it's gone forever. They call it Mueller's ratchet.
Wow.
See, aren't you glad you're hanging out here? Look, this is easy. If you just know if you just let me give you some of the peer-reviewed literature like this, you learn a little bit. You won't have to think about this on your own, because what I'm trying to say is, why don't you let your opponents give you your best arguments? Let your opponents give you the best arguments. If you listen carefully to what they're saying, they're going to give you a gift. Like, there's no effing common ancestor for all proteins. All right? You just have to kind of be sensitive to that, and you'll pick it up. By the way, James Coons contacted me and said, would you be up for a debate with Aaron Ra? And I said, I want to debate the origin of proteins. He said, James came back. He said, I think that's kind of technical. Our audience may not like it. So I called him today. I said, James, it's not my style to deal these shallow talks. Nothing gets settled.
Wait, you mean again? He wants you to go up with Aaron again?
Yeah. And I don't know if Aaron has been asking for me specifically or whatever. James said, you choose the topic, Sal. I said, I want that protein orchard. We're going to argue over it publicly this time. And I said, I'm going to demand a different format. And I think James is like, I don't think that's going to fly over well with the audience. And I said, hey, James, I get it. The modern debate caters to a certain culture and demographic, and that's okay. But I have to protect my reputation and defend and debate topics that I feel I'm going to win. And
to put your best argument out there for the sake
yeah,
of the audience.
Yeah. And I said, if Aaron Rod debates me, I'm going to call him out on it and said, you said to me, there's no effing common ancestor for all proteins. Thank you very much, because now I'll show you why that's problematic for evolutionary theory, starting with collagen.
Understand how? Did James remember that?
I don't think so.
He didn't follow.
Yeah, he probably, because those email chains were so long at some point, I was actually hoping he wasn't
Wait a minute. Did he say that on the debate?
No, he said it in.
That was on. No, I don't think so. I think
No,
he said
the reason I know is he asked me about genetic deterioration. He asked me to give that long list. A list of references.
Okay.
And I hammered him because I gave a long list, and I was just like, dude, I proved my point. You're not even going to acknowledge that. And he said, by the way, what's this about all the proteins not having a common ancestor? I said, why don't you talk to Dr. Dan Stern Cardinal? And he comes back. He said, you're so stupid. Of course there's no effing common ancestor of proteins. That's so obvious. I'm just like, dude, it wasn't so obvious to you until I mentioned it and you had to ask around. And you don't see that that's problematic?
Since that time, when you're on YouTube, sometimes things pop up. And things have popped up with Aaron Ra on the Non Sequitur show specifically, but on a couple of different times he's gone on to the Non Sequitur show. And Aaron Ra is the rudest, the most unscientific, the most verbose. If I would have known that, I don't think I would have debated him in the first place with you. He doesn't use science. He uses intimidation. He uses bullying. He uses insults. He's horrible.
He was nice to you, though, and I
thank him. So I'm just saying.
Yeah.
I don't see much scientific point in debating him. He doesn't have any scientific prowess.
Well, it's going to be interesting if he accepts my terms, because I said it's going to be timed. None of this free discussion except maybe 10, 15 minutes.
And his mic has 2 minutes to be muted.
Right. That's exactly it. 2 minutes for each, or 5 minutes, whatever.
Because
when I start talking, I'm not going to be interrupted.
No, he cannot control himself. He has to be muted.
Yeah. So will he accept my terms? And if not, and if James doesn't want it, I said, James, if you want someone to bore your audience to tears, then you can have me on, because I will resolve this. If I will not let it. I can make my case if we're not going shallow. I'll just cite. I'll make my case. I said I published professionally in this. I'm willing to defend it, because even evolutionists acknowledge it. Even Aaron Ra says there's no effing common ancestor for all proteins. I'm just like, you're already giving a little ground. Don't you realize that, dude?
And you would have to,
I just have to turn down one of my heaters here. Go ahead, Cindi.
Make the connection for, you said he has a certain kind of audience, and it was not a technical audience. So even if you want to make technical points, you'd have to put them in non-technical terms so that people could understand the significance of what you're saying. It took me a long time to understand the whole concept of an orchard instead of a tree. Took me a long time to understand even the significance of Ross saying that.
Yeah. So the thing is that in a short debate, this is what is terrible at a short debate. It's like, am I going to spend half the time educating the audience and then I get cut off in the middle of trying to educate because he's interrupting and he doesn't understand the argument, it's over his head. I just like, that's the problem in a short debate.
Over his head.
Just flew over his head. And Dr. Chris was saying, "Oh, Aaron, you defended this well." I'm just like, dude, what? You're a university professor. You knew what he was saying was just, you did not address the points. So,
well, over his head. That's very interesting because I didn't interpret it like that. I interpreted it as he understood quite well what you were saying, and he was trying to obfuscate it.
I don't care.
Probably true.
Yeah. Well, I don't care at this point. So could we try to keep moving through the video?
Sure.
I think it's a nice pause.
Go ahead.
And yeah.
You made the mistake of asking for my thoughts.
No, okay. So
go ahead.
All right. No, it's all right. We're cool.
Natural selection acts only by taking advantage of slight successive variations. She can never take a great and sudden leap, but must advance by short and sure, though slow steps.
It's really interesting to notice that the more we know about life and the more we know about biology, the more problems Darwinism has and the more design becomes apparent.
Since 1988, Dr. Michael Behe has investigated complex biological systems that seem to defy explanation by natural selection.
For the longest time, I believed that Darwinian evolution explains what we saw in biology, not because I saw how it could actually explain it, but because I was told that it did explain it. In schools, I was taught Darwinian biology. And through college and graduate school, I was in an atmosphere which just assumed that Darwinian evolution explained biology. And again, I didn't have any reason to doubt it. It wasn't until about 10 years or more ago that I read a book called Evolution, a Theory in Crisis by a geneticist by the name of Michael Denton, an Australian. And he put forward a lot of scientific arguments against Darwinian theory that I had never heard before. And the arguments seemed pretty convincing. And at that point, I started to get a bit angry because I thought I was being led down the primrose path. Here were a number of very good arguments, and I had gone through a doctoral program in biochemistry, became a faculty member, and I had never even heard of these things. And so from that point on, I became very interested in the question of evolution, and since have decided that Darwinian processes are not the whole explanation for life.
Michael Behe's skepticism derived in large measure from what modern biology has revealed about life's most fundamental unit, the cell.
In the 19th century, when Darwin was alive, scientists thought that the basis of life, the cell, was some simple glob of protoplasm, like a little piece of jello or something that was not hard to explain at all.
This perception didn't really change too much until the early 1950s. But in the last half century, our knowledge of the cell has just exploded.
Today, powerful technologies reveal elaborate microscopic worlds. Worlds so small that a thimbleful of cultured liquid can contain more than 4 billion single-cell bacteria, each packed with circuits, assembly instructions, and miniature machines the complexity of which Charles Darwin could never have imagined.
At the very basis of life, where molecules and cells run the show, we've discovered machines, literally molecular machines. There are little molecular trucks that carry supplies from one end of the cell to the other. There are machines which capture the energy from sunlight and turn it into usable energy.
There are as many molecular machines in the human body as there are functions that the body has to do. So if you think about hearing, seeing, smelling, tasting, feeling, blood clotting, respiratory action, the immune response, all of those require a host of machines.
When we look at these machines, we ask ourselves, where do they come from? And the standard answer, Darwinian evolution, is very inadequate in my view.
In speaking on the topic of scientific naturalism and evolution...
During the early 1990s, at a series of academic conferences, Behe first shared his doubts about the ability of natural selection to construct complex molecular machines. One machine particularly attracted his attention.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts in all of its glory. It had a propeller and the hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed, you know, that's no chance assemblage of parts.
Behe's reaction was not surprising, for the molecular motors that drive bacteria through liquid each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of machines. It's, you know, it's not convenient that we give them these names. That's truly their function.
Since its discovery, scientists have tried to understand how a rotary motor could have arisen through natural selection. As yet, they have failed to offer any detailed Darwinian explanation. To see why, we must understand a feature of molecular machines known as irreducible complexity.
Cindi, do you have any comments so far? Want to go to the next?
No, we got to stop here. We got to camp here.
Sure.
When we did Ra and Chris, Dr. Chris,
Mm-hmm.
I don't know if you went to Chris's after show, but he was mocking the orchard thing that you were trying to make. What was it? Race or?
Oh, let him mock.
No, but he was saying, "Oh, Sal, all he wants to talk about is." The protein orchard and topoisomerase, as though it's the new bacterial flagellum.
Well, I'll put it this way. It's funny because when Joe DeWeese, you were in that conference with Joe DeWeese. That's kind of what we're saying. This is the new bacterial flagellum, and it's even better.
Well, number one, why is he mocking the bacterial flagellum?
They claim that they have resolved the problem, and there's lots of circular reasoning and obfuscation in that, because the bacterial flagellum has 40 parts, and they said 38 had been, exist elsewhere. Therefore, it can be gradually arisen. And so this is why I said, if you avoid the term irreducible complexity, just as if you say it's a way to find things that are unevolvable, it's okay.
Or all the parts have to be there.
Well, basically,
you take an outboard motor and you take away one of the parts, it ain't gonna work. And they'll say, "Oh, well, yes, it will," but it's not.
Well, one of the problems is they're like 38 parts. 38 of the 40 parts pre-exist, but you still have to assemble it. But the thing is that we found examples where that's not the case. Topoisomerase doesn't have. It's easier to understand. It's very clear. And so, yeah, you could say it's the new bacterial flagellum.
If you say, okay, there's a shaft over there, there's a rotor over there, there's a tail somewhere else, and it doesn't have to always be in that configuration.
Well,
it doesn't take away the fact that that configuration needs all those parts to work the way it works.
But the bottom line is he can say that all he wants. Did he solve the problem? And we sent that letter. We asked for permission to publish it. I said, do you have verified experimental of, the original state, verified, proven, experimentally verified, of how it started, the process that changed it, and how you arrived at that state? And they said, they didn't answer the question. They just evaded and said, well, that's not how science works.
What you were saying about they claim that they have explained how the bacterial flagellum could have arisen incrementally, right? Gradually. But if you look at their explanations, they haven't.
I don't think they've done a good job. I think they gave a good attempt, but what's ended up happening is we have the new generation with better data, and they don't even show up. At least with the bacterial flagellum, they showed up, but with the topoisomerase, they punt. All they could do is mock. They don't actually deliver an explanation, and they know this is problematic, because generally for cellular life of any moderate complexity, without it, it's dead. And something dead doesn't evolve. I mean, it's done,
Well, I'm pretty sure that even without a bacterial flagellum, it would be dead.
No. I know a creationist biologist who demonstrated there are lots of bacteria that had flagellum, and they go into a phase where they don't have one, and they live fine. So, but that's not the case with topoisomerase. That's why topoisomerase is a target of chemotherapies.
Yeah.
So that's why I was excited for you to come to that meeting when Joe DeWeese was talking, and he was talking like an expert in that field, in that protein, because he studied this for 17 years. And you're not going to get some random internet guy or even Aaron Ra challenging someone of that caliber, because he studied it for 17 years. He knows what he's talking about. And he and I co-authored a paper and said, we kind of laugh. It's almost funny. We were citing what the evolutionists were saying. We almost kind of laughed.
Well, I find it stunning that they will attack good scientists like that.
Mm-hmm. Well, that's just par for the course for now. Did you have something else you wanted to explore? Or do you want to watch some more? Because we're getting to the good stuff now.
What was the other main point of that segment? Was it, the fact that we didn't know that the cell was not a blob of jelly, and that there are machines inside of this microscopic cell. Yeah, I mean, the bacterial flagellum is only one of, what did he say? How many machines? One for every function in the body.
Oh yeah, there's so many. You have some that do photosynthesis and all, you know, you need transport and whatever. But we will study cellular biology, God willing, in time.
Mm-hmm.
Okay. Just a FYI, if I may brag, if I can brag some more. One of the researchers there was Jed McCosco. He visited and stayed with our family for, you know, when he had a conference here one time. Michael Behe knows me by name, and so does Paul Nelson. I see Paul Nelson. So these are some of the guys that I know. And William Dembski also knows who I am. I think Stephen Meyer's probably forgotten who I am, but oh well. So let's watch the next segment. So this would be I hope this is chapter 4. If it's a repeat, we'll go to chapter 5.
Yeah.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
The idea of irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function, catching mice. Irreducible complexity also applies to biological machines, including the bacterial flagellar motor.
All told, there are about 40 different protein parts which are necessary for this machine to work. And if any of those parts are missing, then either you get a flagellum that doesn't work because it's missing the hook or it's missing the drive shaft or whatever, or it doesn't even get built within the cell.
In evolutionary terms, you have to be able to explain how you can build this system gradually when there's no function until you have all those parts in place.
The irreducible complexity of molecular machines poses a severe challenge to the power of natural selection. According to Darwin's theory, even very complex biological structures like an eye, an ear, or a heart can be built gradually over time in small incremental steps. Yet, as Darwin made clear, natural selection can only succeed if these random genetic changes provide some advantage to the evolving organism in its struggle for survival.
As I have attempted to show, it is not necessary to suppose that the modifications were all simultaneous if they were extremely slight and gradual. Natural selection is scrutinizing the slightest variations, rejecting those that are bad, preserving and adding up all that are good.
But could Darwin's small, favorable variations have produced a bacterial flagellum? Some scientists doubt the possibility.
How could something new, like a bacterioflagellar motor and all the components that go with it, how could it develop out of a population of bacteria that don't have that system? When each change, according to Darwin's theory, has to provide some kind of advantage.
Imagine such a scenario early in the Earth's history. An evolving bacterium somehow develops a tail and perhaps even the pieces necessary to attach it to the cell wall. Yet without a complete motor assembly, this innovation would provide no advantage to the cell. Instead, the tail would lie immobile and useless, invisible to natural selection, which by definition can only favor changes that aid survival.
The logic of natural selection is very demanding. Unless the flagellum mechanism is completely assembled and actually works, natural selection simply cannot preserve it. It cannot be passed on to the next generation.
The important thing to realize about natural selection is it selects only for a functional advantage. In most cases, natural selection actually eliminates things that have no function or that have a function that harms the organism. So if you had a bacterium with a tail that didn't function as a flagellum, chances are natural selection would eliminate it. The only way you can select for a flagellum is if you have a flagellum that works, and that means you have to have all the pieces of the motor in place to begin with. So natural selection can't get you the bacterial flagellum. It can only work after the flagellum is there and operating.
That was really interesting about it would have to be attached. And that's a trick right there. And even if it's attached, if it's just laying there, you're mute.
Thank you, Cindy. What would I do without you? What would I do without my co-host?
We're both muting on screen.
There now, just as a caveat, there's some subtleties to this that the other side has actually pointed out that there are weaknesses to the argument 30 years ago that we have since shored up. But this is still, it's good to see historically how this has developed to where it is today, because Dr. Chris was right. Topoisomerase is the new bacterial flagellum, but they're going to have a really hard time because it doesn't have it's like the chess game. This is just really devastating. I mean, this is almost like checkmate now. We are playing.
I'm sorry, but the bacterial flagellum has not been debunked, only in their minds.
Yeah. And there's certainly, the topoisomerase, they're not even trying.
Yeah, they're trying to avoid that one.
Right.
You're right. I don't think they could even understand what you were saying.
So just for the viewers, the topoisomerase, the way its role is DNA gets tangled up. If it stays tangled up, you're dead. So the topoisomerase will locate where a tangle is. It will cut one.
Why don't you show that video?
Yeah.
The rope, showing how when the rope gets tangled.
I don't know if I have it handy, but maybe next time, if I remember. Because I'm in the middle of a system overhaul, and my files are all over the place. But what good is it if you have something that just cuts and it doesn't untangle and reconnect? It's just going to shred the genome. A lot of, this is where it's really like, okay, so you're dead. How do you evolve in the next generation? Because you're dead. And there are other systems too along the way that, we're learning more about the chemistry, the biochemistry of life. And we're finding systems like this all over the place.
The machines.
Yeah. Yeah.
And the thing that really stunned me is that it could reconnect, because there's a big difference between reconnecting something that's just a single strand versus reconnecting a wire that has, the plastic sheets surrounding like 12 other wires inside.
Yeah.
Each of those 12 wires has to be connected to the right 12 wire on the other side.
Right.
And isn't a DNA strand like that?
It's
reconnect
two.
A DNA strand.
it's even more complicated than that, because there's lots of, for lack of a better word, Brownian motion. It's shaking around, and it's having to find this, and it doesn't have a lot of active. I mean, if you ask the biochemist to design this without God's template, good luck, because the one thing you can do is you have to, the way you make the machine is you spell it right. If you add the right sequence of amino acids, then you can make a machine. Finding that sequence is not easy. It is brutally difficult, because the way you spell it affects its shape and its function and its operation, and that relationship is not easily figured out. You can't, it's not like in the human world. If you have a piece of wood, you could chisel away and put it in the lathe and shape it the way you want. No, in DNA, with proteins, you have to spell it, and that's very hard. So anyway, let's watch the next segment. We're getting better and better segments the farther we go along. So I think we're at number 5 now.
In 1996, Michael Behe published a book titled Darwin's Black Box. In it, he argued that natural selection, Darwin's designer substitute, could not explain the origin of the bacterial flagellum or any other irreducibly complex biological system. Instead, Behe concluded that the integrated complexity of these systems pointed to intelligent design. Darwin's black box created immediate controversy. Over 75 publications, including some of the world's leading newspapers and scientific journals, reviewed the book. Some scientists praised Behe's work, while others dismissed it as unscientific and religiously motivated. Behe's critics also insisted that he had underestimated the power of natural selection. They argued that the flagellar motor could have been constructed from parts used to build simpler molecular machines, like this needle-nose cellular pump. If the components of the pump already existed, they could have been preserved by natural selection even before the bacterial motor arose. This theory is called co-option.
It's essentially saying that evolution or natural selection at some point was able to borrow components of one molecular machine and build a new machine with some of these components.
Scott Minnick has studied the flagellar motor for nearly 20 years. His research has led him to challenge the co-option argument.
With a bacterial flagellum, you're talking about a machine that's got 40 structural parts. Yes, we find 10 of them are involved in another molecular machine, but the other 30 are unique. So where are you going to borrow them from? Eventually, you're going to have to account for the function of every single part as originally having some other purpose. So you can only follow that argument so far until you run into the problem of you're borrowing parts from nothing. But even if you concede that you have all the parts necessary to build one of these machines, that's only part of the problem. Maybe even more complex, I think more complex, is the assembly instructions. That is never addressed by opponents of the irreducible complexity argument.
Studies of the bacterial motor have indeed revealed an even deeper level of complexity. For its construction not only requires specific parts, but also a precise sequence of assembly.
You've got to make things at the right time. You've got to make the right number of components. You've got to assemble them in a sequential manner. You've got to be able to tell if you've assembled it properly so that you don't waste energy building a structure that's not going to be functional.
Building a molecular machine has been compared to the construction of a house, where workers follow a detailed blueprint and plan for assembly. The foundation of a house is poured before the walls are erected. Plumbing and electrical fixtures are installed prior to enclosing the walls of the structure. Windows must be hung before siding is applied, and shingles are attached only after plywood sheets are nailed to the rafters. So it is with the construction of a flagellar motor.
You build this structure from the inside out. You are counting the number of components in a ring structure or the stator, and once that's assembled, there's feedback that says, okay, no more of that component now. A rod is added, a ring is added, another rod is added, U-joint's added. Once the U-joint's at a certain size and a certain degree of bend, about a quarter turn, that's shut off, and then you start adding components for the propeller. These are all made in a precise sequence, just like you would build a building.
To build a motor correctly requires a complex system of machines that coordinate the timing of the assembly instructions. But how could natural selection construct such a system?
The co-option argument doesn't explain this. You see, in order to construct that flagellar mechanism, or tens of thousands of other such mechanisms in the cell, you require other machines to regulate the assembly of these structures. And those machines themselves require machines for their assembly.
If even one of these pieces is missing or put in the wrong place, your motor isn't going to work. So this apparatus to assemble the flagellar motor is itself irreducibly complex. In fact, what we have here is irreducible complexity all the way down.
We know a lot about the bacterial flagellum. We still have a lot to learn, but we know a lot about it. And there's no explanation for how this complex molecular machine was ever produced by a Darwinian mechanism.
150 years ago, scientists did not know about irreducibly complex molecular machines. Yet Charles Darwin anticipated the difficulty that systems such as these could pose to his theory.
If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down.
Do you like that, Cindi?
I love the quote at the end. Can you read that quote again? My theory would absolutely break down if something could be shown.
Any complex organ could not emerge by slight successive modification. My theory would absolutely break down. You only need one. And Casey Luskin quoted that in Joe DeWeese's premiere of the topoisomerase video.
It seems to me like just about everything is like that.
A lot of things. And the thing that's powerful is it is correct that there is what they call co-option. So you go to Home Depot, you have a lot of the same parts to build different kinds of houses. It doesn't mean that when you have all the parts available, you're going to be able to build what you think you're going to be able to build, unless you have plan and purpose and a design.
And if you've ever tried to construct something and you're in the middle of the project, even if you're trying to bake a cake and you're in the middle of the project and you're missing one ingredient, you have to stop the whole project and go to the store. You need everything in order to make something.
Well, and the other thing is, if you have a recipe, what if you get the steps in a different order? It's going to be nonsensical. You could have a disaster, in fact, it sometimes won't ever make even sense. I mean, you'll realize, doing it in this order is not going to work. So,
Yeah, like putting the roof on first. Now they're going to tell us, well, that's not an animate object. But that doesn't change the point.
Well, the burden is on them. They're claiming natural selection will do it. But I just say, why don't you do what we did. You wrote a letter and you said, do you have verifiable proof?
Well, and I
tried
had
it.
the same exact thought when you and I were talking about, what is it called? Co-option?
Co-option.
That
Co-option
was,
is where you're using parts that exist in another organ. And that's okay. You know, that's okay. But that's why I gave the analogy. Just go to Home Depot. There are lots of parts that are used in different houses in different ways. It
well, not
doesn't
only
mean,
that, but they're already being used because they're part of another organ. What was the name of the thing that they showed at the very beginning? It had a rotor and a shaft, just like the flagellum, but it was called something else. It
a
didn't have
proton pump. Proton pump.
yes.
But it's very
So
similar.
let's say they got some of the parts from the enzymes that make the proton pump or something. Well, that still begs the question, where did the proton pump get them?
Yes. Well, there's no effing common ancestor for those parts, right, Aaron? Now you know. What are the parts? They are proteins. And if there's no effing common ancestor for all the proteins, you got a problem. That's why that was a big deal for him to concede that.
Can you help me make the connection between that and the rotor?
Okay, so there are 40 proteins. The rotor is made of certain kinds of proteins.
Okay, there's 40 protons. No, sorry,
40
proteins that make
40
the bacterial flagellum. Not 40 in the world. 40 that make the bacterial flagellum.
Correct.
Some of them can be borrowed from another organism.
But exactly what you said. Even if you borrow it, you have to keep borrowing from. It has to start somewhere, right?
Where did he get it? Right.
And what do we know? It didn't come from a common ancestor because there is no effing common ancestor for all proteins.
So all the parts are proteins.
Yes. Now you know, does that orchard thing start? Now you're appreciating the significance of how our discussion over, ever since that debate on October 5th and like 6 to 8 weeks of back and forth in the emails. In particularly, R.N. Ross saying there's no effing common ancestor for all proteins.
So, for example, one of the proteins that's in the bacterial flagellum is probably a collagen.
No. Collagens are not in prokaryotes. I don't know. I'm not familiar with the kinds of proteins. The proteins.
To talk about them in those kinds of terms, it helps people understand because we look at them and we see, something that looks like a metal part. We don't imagine it as being a protein that requires a blueprint. Well, you can see even a metal part requires a blueprint, but we don't, yeah.
But you see that the way that it works at that level, very microscopic, nanomolecular level, is the way we do it in the man-made world. We have these milling machines that shape things. We have molds and milling techniques and cutting. In biology, it's almost a lot of it is done just by spelling it correctly in the first place.
Yeah, and our milling machines are all using the same metal to make part, which would make it a lot easier, but they're using inside the cell, they're not using all the same raw material.
Well, the thing is that as you study this more, you'll start to see the significance because it's exactly what you said. Even if it got co-opted, it had to come from somewhere. Where did it come from? And so when we ask, do you have an explanation for the origin of the protein, of the basis of the protein orchard? Because it really was that question. Where did it start? Mm-hmm. Did it just kind of come out of nowhere? What
was the name of that part that wasn't the bacterial flagellum? It was the other one again, proton pump. Okay, so how many proteins are in the proton pump?
I don't know. I'm sorry. I don't know.
Obviously, it would be less than 42, right?
Yeah, I think it might be 13. It's not as complicated.
Well, that means, let's say there's 13 and there's 42 in the flagellum. Well, where did the other 20 some come from?
From other parts.
Oh, I see. Mm-hmm.
But there are at least 2 that have what they call no homologue. But even then, as I said, Scott Minnick in that video made the point. He said it's the assembly instructions he thinks are actually more complicated.
Not only that, but you have to have a foreknowledge of what you're building and what it's for.
So all right, let's say I gave you all these ingredients that are in your pantry. Is it going to automatically become a gingerbread thing like you cooked up? No. You need
not only that, but how would you even know that a raw egg, baking soda, baking powder, buckwheat, and pumpkin could become bread?
Right. You need foresight. And there is, by the way, a world-class chemist who buddies around with the Nobel Prize winners. His name's Marcos Eberlin. He wrote the book Foresight. It's exactly what you said. He recognizes the same problem. So we're having a lot of elite scientists say, guys, evolutionary biology doesn't seem to jive with what we know about chemistry and physics. That's a problem. So
these non-elite evolutionary biologists don't listen to these elite evolutionary biologists.
So again, I think the thing about assembly, I don't think we even understand how assembly. We're only beginning to understand what they call protein regulation, because as Scott Minnick said, you have to do this in the right sequence. We're only learning how difficult this is to get things in the right sequence if you're going to build something complicated. So, we'll just be patient we'll be patient. We're going to figure out better and better examples of what's not going to work, and where Darwin's theory will absolutely break down. So let's get to the next episode.
Okay.
And I think we just finished part 6. Let me just verify that we did. And then if we did, we'll go to part 7, because this is getting better and better each time.
There are really 2 big questions in biology. How do you get new living forms with new structures like wings and eyes from life that already exists? And secondly, how did life originate on Earth in the first place? Now, of course, we know that Darwin spent most of his life formulating an answer to the first of these 2 questions.
Charles Darwin compared the history of life on Earth to a great branching tree. The base of the tree represented the very first living cell, and the branches were new and more complex life forms that had evolved over time from the first primitive organism.
Darwin was trying to explain how the branches on the tree of life originated. He was trying to show how natural selection could have modified existing organisms to produce the great diversity of plant and animal life that fills the earth today. But when it came to the base of the tree, which represented the origin of the first life, the first living cell, Darwin had very little to say. In fact, in The Origin of Species, he didn't even address the question of how life might have originated from non-living matter.
The only glimpses we have of Darwin's opinions on the subject appear in a letter he wrote to a colleague named Joseph Hooker.
regarding the first production of a living organism. If, and oh what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat and electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes, at the present such matter would be instantly devoured. But this may not have been the case before living creatures were formed.
During the final years of his life, Darwin did little to develop his idea that a primitive cell might have emerged from simple chemicals in the primordial waters of the early Earth. But later in the 1920s and 30s, a Russian scientist named Alexander Oparin formulated a detailed theory about how this could have happened. It was called chemical evolution.
Oparin thought that he could explain the origin of the first life using Darwinian principles. He envisioned simple chemicals combining and recombining to form larger molecules, and then these larger molecules organizing themselves with the help of chance variations and natural selection into the first primitive living cell.
Over the next 3 decades, many scientists worked to develop and refine these ideas as they pondered the questions both Oparin and Darwin had raised. How could life have evolved from simple chemicals? One man thought he knew.
The problem of biological origins has, for a very long time, I would say, has been a real deep interest to me just because of the scale of the problem, the importance of it. Where did we come from? Why are we here? All that kind of question. Probed from the point of view of natural science.
During the late 1960s and throughout the 70s and early 80s, Dean Kenyon was one of the leading chemical evolutionary theorists in the world. And like others in his field, he was trying to explain how life on Earth began through a purely natural process.
In 1969, Kenyon co-authored an important book on the origin of life.
Gary Steinman and myself thought that if we were to pull together in all of the lines of empirical evidence that had accumulated by the mid to late 60s in one continuous argument, we were very enthusiastic about the possibilities for explaining the origin of the main life-building elements.
I'll go to the next segment because this is now you've met Dean Kenyon, and we'll just continue because now it's really getting interesting. This is the best, to me, this is going to be the best parts of the whole series.
You know, when I used to teach this in the Science Center,
Mm-hmm.
people would say, oh, nobody believes that. When I would teach about the primordial soup and the electricity and the chemicals, well, he just said it, and he just said that Darwin said it.
See?
And they just deny things that sound stupid.
Well, things are changing. You know, it was so bad that in today they did change the story with this RNA world, but it was the prevailing theory for a long time.
Not only that, where'd they get the water? Where'd they get the phosphorus salts? Where'd they get any of it? And how is chemicals floating in water going to arrange themselves into a cell with a cell wall? They're just going to automatically go into a wall that has things inside of it?
Well,
like a nucleus.
Yeah. Well, that'll get into more advanced cellular theory, but maybe we should just keep rolling because this is really getting good. If that's all right. Okay.
Despite his optimism, Kenyon faced a significant problem. To explain how life began, he first had to account for the origin of the essential building blocks of every cell that has existed on Earth. Large, complex molecules called proteins.
Proteins have a wide range of function in the cell, everything from structural requirements in terms of scaffolding of the cell.
Of every cell that has existed on Earth, large complex molecules called proteins.
Proteins have a wide range of function in the cell, everything from structural requirements in terms of scaffolding of the cell, the cytoskeleton, to enzymes, where they're actually processing molecules to harvest energy or to build components of the cell.
Proteins do pretty much all of the jobs inside of the cell, except for storing genetic information. That's left to the DNA, the RNA. But all the day-to-day jobs, cleaning up the cell, making energy, it's all proteins.
Kenyon knew that proteins would have been as important to the first life as they are to living cells today. He also recognized the complexity of their construction.
By the 1960s, scientists had determined that even simple cells are made of thousands of different types of proteins. And the function of these molecules derives from their highly complex three-dimensional shapes. The irregular shapes of some proteins allow them to catalyze or trigger chemical reactions because of the hand and glove fit that they have with other molecules in the cell. While other protein molecules form interlocking structural components.
The individual parts of a bacterial motor, like this ring structure, are each made of either a single protein molecule or an assembly of proteins fitted together into a specific shape. These proteins are, in turn, made of smaller chemical units called amino acids that are linked together in long chains.
There is a very great degree of intricacy of architecture down in the cell units in these protein-forming amino acids.
In nature, 20 different types of amino acids are used to construct protein chains. Biologists have compared them to the 26 letters of the English alphabet.
Alphabetic letters can be arranged in a huge number of possible combinations, and it's the sequential arrangement of the letters that determines whether you have meaningful words and sentences. If the letters are arranged correctly, you'll get meaningful text. But if they're not arranged correctly, you'll get gibberish. And the same principle applies for amino acids and proteins.
There are at least 30,000 distinct types of proteins, each made of a different combination of the same 20 amino acids. They are arranged, like letters, to form chains, often hundreds of units long. If the amino acids are sequenced correctly, then the chain will fold into a functioning protein.
Proteins are arranged with their amino acids in such a way that the amino acids collapse on each other into an architecture that is pre-programmed by the order of the amino acids. It folds into a certain structure, and that structure can do a certain function. So all proteins in the cell have a certain three-dimensional pattern that's based on the arrangement of amino acids in the chain.
This arrangement is critical, for if the amino acids are incorrectly sequenced, a useless chain forms, and instead of folding into a protein, it will be destroyed in the cell.
Proteins, like written languages or computer codes, have a high degree of specificity. The function of the whole depends upon the precise arrangement of the individual parts.
But what produces the precise sequencing of amino acids that gives rise to the specific shapes and functions of proteins? During the 1950s and 60s, discoveries about protein structure forced biologists to confront this mystery. Dean Kenyon believed he could solve it. In his book, Biochemical Predestination, Kenyon and his co-author, Gary Steinman, proposed an intriguing theory. Kenyon wrote, "Life might have been biochemically predestined by the properties of attraction that exist between its chemical parts, particularly between amino acids in proteins."
At the time that biochemical predestination came out, I and my co-author were totally convinced that we had the scientific explanation for origins.
Kenyon proposed that the chemical properties of the amino acids caused them to be attracted to each other, forming the long chains that became the first proteins, the most important components in the living cell. And this meant that life was effectively inevitable, predestined by nothing more than chemistry.
Many scientists embraced Kenyon's ideas, and over the next 20 years, Biochemical Predestination became a best-selling text on the theory of chemical evolution. Yet 5 years after the book's publication, Kenyon quietly began to doubt the plausibility of his own theory.
It was during that whole period of time that my doubts about certain aspects of the evolutionary account became apparent. When coming into contact with a powerful counter-argument given to me by one of my students, and I could not refute that counter-argument.
Isn't that intriguing?
So a student, probably a creationist.
Yes, and I know the name of the book. I found out from another video. It's a book, Cybernetic Approach to Evolution by A.E. Wilder Smith. I have that book.
You mean that is the student?
No, the student bought a book.
Oh.
It was written by A.E. Wilder Smith, who has 3 PhDs, one of them in chemistry from Oxford, I think, or one really good school. 3 PhDs. He was also a NATO general. Talk about someone who's overachieving. And A.E. Wilder Smith was a famous creationist. He was a very previous generation. This would be in the 1960s and 70s. So this is why this is just a great this is a great video, because it ties, it even reaches back into the depths of creationism that even preceded Henry Morris. You know, it was a guy from England, which is A.E. Wilder Smith, and so this is this lineage of creationism that's tied even with, the pagan notions of intelligent design. That's the modern day version of intelligent design.
I just love thinking that a student,
yes,
like me, would be in class, raise their hand, ask a question, and make a big change in the world.
Yes. And in this case, it was giving the professor a book, but he was open. And I think you saw a video on my channel about Richard Lumsden. That was a student. And you may have played it on your
student?
No, it was a different story. But that was the case.
He was changed by another student also. Yeah, I have his testimony. I uploaded it from your channel.
Yeah. See? But that was I have to venture that someone was praying hard. And Richard Lumsden said he realized it was his daughter that was praying very hard for him.
Oh.
Because his daughter became a Christian and then started praying for her father.
Yeah.
Yeah.
And what was the point at the very beginning of that video? That was the tail end of it, but what was at the beginning of it? It was something good.
Of this segment
Yeah.
It was about biochemical predestination. Oh, okay. So you have proteins. You can't really have a functioning cell without proteins. And because they do all the essential parts, all the essential functions. And at the very beginning, I thought I saw a heart-shaped thing. It looked like a topoisomerate. It's very close to a topoisomerate.
I thought so too.
Huh?
I thought so too.
There you go.
Well, and just a reiteration of, okay, I'm slowly getting this. Proteins are comprised of amino acids.
And how many?
And if the amino acids are 20, and they're alphabetic, and they're combination like words. Like if you have an A and a B. Anyway, what I was getting out of that was the proteins are comprised of these long chains of amino acids. And not only does that chain have to be in the right order, but God made it so that when it folds in a certain three-dimensional shape. I mean, if you have a chain, you can fold that chain in any number of shapes. But that the fold itself is part of the design. It's mind-boggling.
Yes.
The specificity, the complexity. I mean, it's.
Yeah. Are you enjoying your molecular biology, your exploration of this? I hope it's.
Yeah, it's really.
I hope this is opening kind of, new perspectives and avenues for your understanding of God's creation.
Mm-hmm. And the whole point started out in this segment about turning a chemical into a cell, and that even if you could get the shapes right, because they're saying chemicals have attractions one to another, and so predestined by attraction. Well, where did that come from, first of all? And second of all, you can have the cell in the right shape, even with all the right components, but you still got to have life.
Okay, so one thing I need to point out, in the next segment, you'll find out Dean Kenyon realized there's not this inherent attraction.
Mm-hmm.
It's actually, there's no inherent tendency to form a protein. He thought for sure, just like when you mix oxygen and hydrogen, you throw a spark at it, you get water explosion,
Mm-hmm.
that it would arrange itself spontaneously. He found out that's not the case. These are it obeys kind of different chemical principles because this is partly organic chemistry.
Interesting. And I've heard with water, it's quite the opposite, that the natural tendency is for it to not form water. And so it's it's kind of a miraculous part.
Well, no, it doesn't. Well, this is the amino acids will not spontaneously polymerize in water. So yeah, that's another thing. So anyway,
the H and the 2 and the O, you can't just throw electricity at it and have it naturally turn into water either. It wants to not be water.
Oh, I think it does. That's why it's a very powerful reaction, and that's what powers parts of the early spaceships like the Saturn V. There were stages where they just mixed hydrogen and oxygen. So they had hydrogen and oxygen, and they would have an igniter, and that thing would just.
Is that fusion or fission?
No, that's just a chemical reaction.
So it gives off energy.
Yeah.
Interesting.
Anyway,
go ahead.
Okay. So I think we were at chapter 8, and let's go to 9. Hopefully I didn't skip. We'll find out if I accidentally skipped.
Yet Kenyon realized that he faced a narrowing set of options. By the 1970s, most researchers had rejected the idea that the information necessary to build a
I think I skipped one. I'm sorry, Cindy. Let's try with segment 8. I was getting so ahead of myself. Anyway, this is getting really good.
Kenyon was challenged to explain how the first proteins could have been assembled without the help of genetic instructions. In living cells today, chains of amino acids are not formed directly by forces of attraction between their parts, the scenario Kenyon envisioned on the early Earth. Instead, another large molecule within the cell stores instructions for sequencing the amino acids in proteins. It is called DNA.
Initially, Kenyon believed that proteins could have formed directly from amino acids without any DNA assembly instructions. And that's why so many scientists were excited about his theory. But the more he and others learned about the properties of amino acids and proteins, the more he began to doubt that proteins could self-assemble without DNA.
In DNA, Kenyon encountered a molecule with a property he could not explain through natural processes. For locked securely within its double helix structure is a wealth of information in the form of precisely sequenced chemicals that scientists represent with the letters A, C, T, and G. In a written language, information is communicated by a precise arrangement of letters. In the same way, the instructions necessary to assemble amino acids into proteins are conveyed by the sequences of chemicals arranged along the spine of the DNA. This chemical code has been called the language of life, and it is the most densely packed and elaborately detailed assembly of information in the known universe.
Like other scientists working on the origin of life, Kenyon realized he had two choices. Either he had to explain where these genetic assembly instructions came from, or he had to explain how proteins could have arisen directly from amino acids without DNA in the primordial oceans. And in the end, he realized he could do neither.
It's an enormous problem how you could get together in one tiny submicroscopic volume of the primitive ocean all of the hundreds of different molecular components you would need in order for a self-replicating cycle to be established. And so my doubts about whether amino acids could order themselves into meaningful biological sequences on their own without pre-existing genetic material being present just reached, I guess for me, the intellectual breaking point sometime near the end of the decade of the 70s.
As Kenyon reevaluated his theory, new biochemical discoveries further weakened his conviction that amino acids could have organized themselves into proteins.
The more I conducted my own studies, including a period of time at NASA Ames Research Center, the more it became apparent that there were multiple difficulties with the chemical evolution account. And further experimental work showed that amino acids do not have the ability to order themselves into any biologically meaningful sequences.
Faced with mounting difficulties in his own theory and a growing body of scientific data about the importance of DNA, Kenyon was forced to confront the absolute necessity of genetic information.
The more I thought about the alternative that was being presented in the criticism and the enormous problem that all of us who worked on this field had neglected to address, the problem of the origin of genetic information itself, then I really had to reassess my whole position regarding origins.
For Dean Kenyon, a new question became the focus of his search for life's origin. What was the source of the biological information in DNA?
If one could get at the origin of the messages, the encoded messages within the living machinery, then you would really be on to something far more intellectually satisfying than this chemical evolution theory.
I'm just going to move to the next segment to complete the thought.
Yet Kenyon realized that he faced a narrowing set of options. By the 1970s, most researchers had rejected the idea that the information necessary to build the first cell originated by chance alone. To understand why Consider the difficulty of generating just 2 lines of Shakespeare's play Hamlet by dropping Scrabble letters onto a tabletop. Then consider that the specific genetic instructions required to build the proteins in even the simplest one-celled organism would fill hundreds of pages of printed text.
Of course, serious origin of life biologists did not believe that life had arisen by chance alone. Instead, they envisioned natural selection acting on random variations among chemicals to produce the first life. But there was a problem with this proposal.
By definition, natural selection could not have functioned before the existence of the first living cell, for it can only act upon organisms capable of replicating themselves, cells equipped with DNA that pass on their genetic changes to future generations.
Without DNA, there is no self-replication. But without self-replication, there is no natural selection. So you can't use natural selection to explain the origin of DNA without assuming the existence of the very thing you're trying to explain.
Chance, natural selection, and his own theory of self-organization had all failed to explain the origin of genetic information. Now Kenyon saw only one alternative.
We have not the slightest chance of a chemical evolutionary origin for even the simplest of cells. So the concept of the intelligent design of life was immensely attractive to me and made a great deal of sense, as it very closely matched the multiple discoveries in molecular biology.
Cindi, are you there?
Yeah.
Hope you're enjoying it.
Holy cow.
Isn't that a beautiful story? Aren't you glad we're doing this? Now, let's go to chapter 10.
How can they assemble themselves without instructions?
A recipe.
And
Okay.
I've also heard evolutionists say that DNA is not an instruction and that it's not a language. What the heck?
Yeah. A rose is a rose by any other name. If they don't want to use that term, fine. How about the DNA has to be organized for this correct, the DNA has to have some level of organization for the cell to exist. And if it's an improbable organization, it's done. So I try to avoid the semantic games of saying it's a code, it's not a code. It's like, it doesn't matter. It's organized in a way that makes the cell alive.
Well,
what's the source of the organization? Easy.
I'm not even talking about organization. I'm talking about it is instructing the protein how to construct. They can't assemble the amino acids into a functioning protein without something telling it where to attach.
Okay, so you don't have to use the word instruction. The organization of DNA is the sequence that ends up being like a recipe. Whether you say it is a recipe or like a recipe, doesn't matter. If you don't have the DNA having the right sequence of ACTG, it fails. So I'm just trying to give you a little pointer how you can avoid all their they're doing a lot of obfuscation, semantic games.
I guess that was what I was trying to inquire.
You can cut through it. If you avoid the word instruction, code, or language, and just say you have to have certain sequence. Most sequences will fail to make life. Only a highly improbable sequence will succeed. That's the cutting the chase. You'll be able to.
And if the amino acids cannot arrange themselves without a plan, an instruction, an implementer, then why can't you call it an instruction?
I'm right there with you.
But
but I'm just saying, if you're
dragon.
Okay, but if you're in polemics and in a debate, you could say call it whatever you want. It doesn't change the fact anyone invoking naturalism has a problem. You see, this is the game that will be played. If they can't really win on the arguments, they do these little silly maneuvers to try to derail. Remember, it's all about derailing. It's not about getting to the truth.
Basically,
for them,
the amino acids aren't going to form any meaningful protein if there's no DNA, regardless of what you want to call it.
It's not going to form certainly complex proteins that are required for life.
Right.
I wouldn't say any, but it's not going to form anything like a topoisomerase or a homohexameric helicase or tetrameric helicase, a potassium ion channel. It's not going to happen. So let's go to segment 10. I'm sorry.
I just said go ahead. This is awesome.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
All right, Cindi, are you there?
Sorry, it's getting late over here. I'm brushing my teeth and getting ready for bed while I'm listening.
Okay,
So
there's
I
only
forgot
one.
about
There's
another
only,
level of complexity. Once the whole thing is constructed, then it has to know where to go
yeah,
and what to do.
yes. Oh, we can explore all that. Okay, so we only have one more 5-minute segment, but since this is about molecular biology, you saw it in a nutshell. So how about I walk it through one more time, that same segment, and I'll pause it and just talk, and
Okay.
and then we'll finish up. We're almost at the end, but I'm just going to replay this, but I may pause it in the interim. So just give me one. Let's try this again.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work.
Okay, I have to pause here. This is a eukaryotic cell. Just be careful when you show this segment because the other side will say, oh, it wasn't that complex. Just a little caveat. Let's just go on.
After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule.
Okay, so he used the word transcription. So the first part of the central dogma of molecular biology is DNA often transcribes to RNA. So we'll just continue.
Another machine then copies these instructions to form a molecule known as messenger RNA.
Okay, so that class of RNA, they call it messenger RNA. So you can just say RNA, but if you say messenger, that's more specific. So the central dogma, you have the first half of the central dogma right there. DNA transcribes to RNA.
When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins.
Did you hear that? Translation. RNA translates to proteins.
Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
I'm going to the
No human
Huh?
being, no human being could think of that.
Yeah. And he said it's the most compelling evidence of intelligent design. That's why I love the molecular level. It's powerful. Now let's go 11 of 12. I'm sorry, there's actually one more after this. We're almost done.
When I look at molecular machines or the incredibly complex process by which cells divide, I want to ask, is it possible that these things had an intelligence behind them, that there was a plan or a purpose to this structure? Science ought to be a search for the truth about the world. Now, we shouldn't prejudge what might be true. We shouldn't say, I don't like that explanation, so I'm going to put it to one side. Rather, when we come to a puzzle in nature, we ought to bring to that puzzle every possible cause that might explain it. One of the problems I have with evolutionary theory is it artificially rules out a kind of cause even before the evidence has a chance to speak. And the cause that's ruled out is intelligence.
Since the late 19th century, since the time of Darwin, in fact, in part because of Darwin's writing in The Origin of Species, scientists came to accept a convention, a definition of science that excluded the possibility of design as a scientific explanation. And that convention has a name. It's called methodological naturalism. And it just means that if you're going to be scientific, you must limit yourself to explanations that invoke only natural causes. You can't invoke intelligence as a cause. And yet, curiously, we make inferences to intelligence all the time. It's part of our ordinary reasoning to recognize the effects of intelligence.
Consider, for example, these hieroglyphic messages carved upon the ruins of Egyptian monuments. No one would attribute the shapes and arrangements of these symbols to natural causes like sandstorms or erosion. Instead, we recognize them as the work of ancient scribes, intelligent human agents. Similar reasoning leads us to conclude that the mysterious stone figures on the shores of Easter Island were not formed by the actions of wind and water over great periods of time. Nor do we presume that plants could grow into these familiar shapes without some manner of intelligent guidance.
Of course, we make these inferences all the time, and we know they're correct. But the question is, on what basis do we make these inferences? What are the features that enable us to recognize intelligence?
Recently, in a book titled The Design Inference, mathematician William Dembski has made an important breakthrough in understanding design reasoning. Dembski has identified the specific features of artifacts that cause us to recognize prior intelligent activity.
I came to this by trying to look at how do we reason about design? What are the logical moves that we have to go through in order to come to a conclusion of design? So what I'm trying to do is to establish reliable, empirical, scientifically rigorous criteria for deciding whether something is in fact designed. So I was looking at the logic of it. And what I found was you need improbability and you need specification, the right sort of pattern, these objective patterns.
According to Dembski, human beings correctly detect the activity of intelligence whenever they observe a highly improbable object or event that also matches a recognizable pattern. Just such a pattern is found in the Black Hills of South Dakota.
If you travel through the West, you'll see lots of different shapes on mountainsides, most of which mean nothing at all. They're just rocks strewn in various patterns. But what you don't see are the faces of Lincoln, Jefferson, Teddy Roosevelt, and George Washington on mountainsides. The only place you see that is in South Dakota. And the reason it's there is because a sculptor, an eccentric sculptor, decided that he wanted to honor these presidents by spending the larger part of his life chiseling their faces in the side of that mountain. That pattern is improbable. A random hillside is also improbable, but a random hillside doesn't specify anything. We do know, though, that there were 4 guys who were presidents of the United States who had particular patterns with their faces, and those patterns on the mountainside in South Dakota match faces elsewhere.
If I look up and see the faces, I immediately recognize that they match the faces of the 4 presidents that are known from money or portraits at the National Gallery or paintings and books. And so I realized when I look at Mount Rushmore, we have not only a highly improbable configuration of rock, but one which matches an independently given pattern that reliably indicates intelligence.
So we have a small probability, specification, its design.
On a seashore, another improbable pattern etched into the earth illustrates how we detect design. No one would infer that this message was written by the movement of the tides. Instead, because of the characteristics of this pattern, we identify the words as the products of intelligence.
That improbable arrangement also conforms to an independently given pattern, namely the shapes of the letters that we recognize from English alphabet and the words that we know from English vocabulary. And so it's the improbability of the arrangement plus the fact that it conforms to an independently given pattern that triggers the awareness of design.
This illustration suggests that William Dembski's criteria for design detection, small probability and specification, are essentially equivalent to information. The type of information present not only in pictures, written texts, and numeric sequences, but also encoded in software and radio signals. The ability to detect information in electromagnetic transmissions has made possible a unique search for intelligence. For more than 3 decades, astronomers involved in SETI, the search for extraterrestrial intelligence, have monitored radio signals from outer space in an attempt to find information-rich patterns. Typically, radio telescopes receive either random noise or simple repetitive signals, produced naturally by stars, galaxies, and other celestial objects. But astronomers recognized that if they ever identified an information-bearing signal, it would confirm the existence of intelligent life beyond the Earth. Some have speculated that an extraterrestrial civilization might have attempted to communicate by transmitting messages in the universal language of mathematics, perhaps through a recognizable pattern, like a series of prime numbers.
You're not going to get that by chance. So you need complexity or improbability, lots of prime numbers, and you also need a pattern. And it has to be the right sort of pattern. It's not a pattern that you're imposing. It's a pattern that's there objectively.
To date, SETI research has failed to detect any pattern or information that would indicate intelligence in a distant galaxy. But in another universe, much closer to home, scientists have discovered a wealth of information within the nucleus of the living cell.
DNA has a structure that is ideal for carrying information. In the A's, T's, C's, and G's, the bases of the double helix of DNA, is the potential for storing a tremendous amount of information.
There is, in fact, no entity in the known universe that stores and processes more information more efficiently than the DNA molecule. A full complement of human DNA has 3 billion individual characters. Analysis of the DNA molecule's coding regions show that its chemical characters have a specific arrangement that allows them to convey detailed instructions or information, much like letters in a meaningful sentence or binary digits in a computer code.
Bill Gates has said that DNA is like a computer program, only much more complex than any we've been able to devise. And if you reflect on that even for a minute, it's a highly suggestive observation, because we know that Bill Gates does not employ wind and erosion or random number generators to generate his software. Instead, he employs intelligent engineers, software engineers. And so everything we know in our experience suggests that information-rich systems arise from intelligent design. But what do we make of the fact that there is information in life, in every living cell of every living organism? That's the fundamental mystery. Where does that information come from?
And now the last segment. I'm going to the last segment, and I know both of us are sleepy, so we'll just finish it out since we're at the end.
I can't wait to upload this on my channel.
Isn't this beautiful? Be sure, yeah, it's going to be good. You might have to stitch pieces together, but it's great.
For the past 15 years, philosopher and scientist Stephen Meyer has worked to answer this question. Meyer has developed an argument to demonstrate that intelligent design provides the best explanation for the origin of information necessary to build the first living cell.
The information that the DNA molecule holds. It's part of our knowledge base that intelligent agents can produce information-rich systems. So the argument is not based on what we don't know, but it's based on what we do know about the cause and effect structure of the world. We know at present there is no naturalistic explanation, no natural cause that produces information. Not natural selection, not self-organizational processes, not pure chance. But we do know of a cause which is capable of producing information, and that is intelligence. So when people infer design from the presence of information in DNA, they're effectively making what's called in the historical sciences an inference to the best explanation. So when we find an information-rich system in the cell, in the DNA molecule specifically, we can infer that an intelligence played a role in the origin of that system, even if we weren't there to observe the system coming into existence.
Meyer's work on the origin of genetic information is now part of a comprehensive scientific case for design that grew out of a meeting of scientists and philosophers on the central coast of California in 1993. Their objective was to reassess an idea that had dominated biology for more than a century. In the process, they gave birth to a theory that has become known as intelligent design.
To me, the great promise of design is it gives us a new tool and explanation that belongs in the toolkit of science. Intelligent causes are real, they leave evidence of their existence, and a healthy science is a science that seeks the truth and lets the evidence speak for itself.
The argument for intelligent design is based on observation of the facts. Now that's my definition of good science. It's observation of the facts. Now when you observe the facts, as Michael Behe has done, what do you observe? You observe this incredible pattern of interrelated complexity.
And the way we conclude intelligent design for the bacterial flagellum is the same way we conclude intelligent design for an outboard motor. When we see an outboard motor, we see the way the parts interact and so on. We know somebody made that. The reasoning is the same for biological machines. So the idea of intelligent design is a completely scientific one. Certainly it might have religious implications, but it does not depend on religious premises.
When I look at the evidence objectively, without ruling out the possibility of design, design just leaps up as the most likely explanation. And that's why I believe that it's true.
I think design is back on the table. You know, we can't explain these systems by natural law. And if we're searching for truth, and they are in fact designed, if we have to be design engineers to understand them, then I say, what's the problem? You know, you go where the data leads you. And the implications, yeah, they have profound metaphysical implications, but so be it.
So it's a powerful idea that the universe is rational and comprehensible, underwritten by a supreme intelligence that meant for this world to be understood, is something that underwrites then the program of science, because then you can go out and look at the world and the world will make sense. If it's all just a chaotic assemblage, there's no reason to expect any rationality out there. But if it in fact is the product of a mind, then you can go out and science becomes this enormous, wonderful puzzle-solving project in which you can expect to find rationality and beauty and comprehensibility right at the foundation of things.
150 years ago, Charles Darwin transformed science with his theory of natural selection. Today, that theory faces a formidable challenge. Intelligent design has sparked both discovery and intense debate over the origin of life on Earth. And for a growing number of scientists, it represents a paradigm, an idea with the power to, once again, redefine the foundations of scientific thought.
During the 19th century, scientists believed that there were two fundamental entities, matter and energy. But as we enter the 21st century, there's a third fundamental entity that science has had to recognize, and that is information. And so as we encounter the biology of the information age, the suspicion is growing that what we're seeing in the DNA molecule is actually an artifact of mind, an artifact of intelligence, something that can only be explained by intelligent design.
Wow. You're muted.
I'm sorry. I hope you had fun. Thank you for hanging with me for like 2 hours and almost 45 minutes. I hope it was worth.
I'll edit it down before I put it up on my channel. But I can't wait to get it up there.
Well, you know, you don't have to have our conversation. If you just get the Illustra Media pieces, they're about 12 pieces, stitch them together, it'll be about an hour and 6 minutes.
Yeah. I'll just take out the part where it's the repeat of the intro. I like some of our comments though, but I'll go through it. I hope in my spare time. Editing is so time consuming.
Well, you know what? I could probably stitch it together for you. All the Illustra parts.
You don't want our comments in there?
Well, it'll be a lot shorter.
So the Illustra by itself is going to be over an hour, is what you're saying?
About an hour and 6 minutes, I think. Hour 10. It's not, you know, this whole thing was 2 hours 42 minutes, but we had some fun comments. We were talking about Aaron Ra and Dr. Chris. That was kind of fun.
I could break it into 2 videos.
Yeah. My favorite part was Dean Kenyon. You see, and there's also, I'll give you some backstories. I don't know if he was a Christian before he started accepting intelligent design, but I'm pretty sure afterward he was. Pretty sure. He hung around certain circles that were creationist and Christian. So I'm pretty sure he became a Christian if he wasn't already. sadly, I think, he was very elderly, um, toward the production of this. I don't know if he's still alive, but God rest his soul.
If
If
he was
he's
a Christian, he's still alive.
Yes, amen. Then the other thing at the very end, Phil Johnson, who organized the conference, he's the one who financed a lot of the intelligent design movement because he was a law professor at
Wow.
Berkeley. He had the money. He relates.
Berkeley?
Yeah,
Why was he not fired?
because he was a law professor, not a scientist.
Oh my gosh, they must not have known what he was doing in the background. Berkeley is the most liberal place in this country.
Well, remember, this is 1993. It was a different era. They were probably more tolerant then.
Yeah.
But the story is he got the hold of that book, Evolution, a Theory in Crisis, which also affected Michael Behe. Phil Johnson read it, rejected evolution, and then rededicated his life to Christ. He became a Christian. So even though they didn't mention, a lot of what we would expect in creationist talks where they talk about the Bible and creation, there was a lot of people who became, it affected them. One of the people there, Paul Nelson, is a young earth creationist.
Does he work DI?
Yes.
He works with DI?
Yes. And Joe DeWeese is a young earth creationist. So there are at least 2 young earth creationists with the Discovery Institute.
Okay.
At least 2. There could be more.
That's good.
Yeah. So, and if I may say, I'm really glad you're getting, I got to tell you, these guys are great. They're just wonderful human beings, and
you get a sincere spirit from them. But I have to tell you, Sal, I know you're about to say you're glad I'm getting a better creationist education, but guess what? All this stuff is in the Science Center. A lot of it is in the Science Center. And I saw that's where Kent plagiarized his ideas. I saw a lot of this stuff before, but it's good to see it afresh because he doesn't emphasize that stuff. He emphasizes more of the dinosaur and the geography. Which I
didn't. I hope you, well, even if it was, I hope you've gained a new appreciation for what was there, because this stuff really does carry the day for people that are in the sciences like myself.
Yeah. Well, and like you said, different people get convinced by different things. Those men were convinced by those books. I think when I upload this, I will put images of those books at the end too.
Yeah. Well, I'm going to close the recording. We can meet a few minutes backstage after the recording. I have a special song for you.
Oh, thank you, sweetheart.
So I'd like to say, let me close in a prayer.
Okay.
Lord, your majesty and glory are revealed in the things you have made.
Mm-hmm.
We stand in awe of you, and we honor you in Jesus' name. Amen. And so along those lines, the song I'm going to play.
Hey,
yes.
That was the first thing that I was thinking when you turned it off for the last time. There is no excuse. And I know that sounds harsh. And I've heard, I think it was Michael McKinnon Mitchell. He, in his documentary Against Kent, he said, yeah, Romans 1 is just not enough. Like, yeah, it's enough just to see creation and bam, you're supposed to accept it. He was mocking that.
Yeah, and what do they have? What do they have to offer? Except we don't know. Because you ask them, how did it evolve? And they say, we don't know.
Well, and
right?
the more you look into it, I mean, I don't know what it is, why we look at it and we see God, and why they look at it and they don't. But that was my first thought, was there's no excuse. There is no way that happened by chance, from nothing. Even give them the primordial soup, it still isn't going to happen.
Okay, you asked. I'll give a scripture that gives. This would be my answer. Jesus said, "But your eyes are blessed because they do see, and your ears because they do hear."
Yeah.
So you are blessed that you could see when other people are blind.
Spiritually dead.
So, rather than me worrying about them, I'm just going to be grateful. Your eyes were open and my eyes were open before it was too late.
Yeah.
Amen.
I do get confused if that's the way it works. Why doesn't he just open everybody's eyes? But then that's just Romans 9, it's his game.
Yeah.
It's not a game. It's his creation.
Yeah.
It's who he wants to choose.
And it related to Pharaoh's heart being hardened. And I've tried to tell some people, don't get too upset anymore, because God was glorified because he kept hardening Pharaoh's heart. Sometimes their hearts are hardened because God ordained it. And just,
Yeah. I don't concern myself with matters far beyond my reach.
Yeah. So just be grateful. Your eyes are blessed because they do see, and your ears because they do hear. And that's enough for today. So how about we finish, and then we'll talk a little bit backstage.
Okay.
All right.
Let's pray, Sal.
Okay, 1 second. It's queuing up, and I'll stop it. All right. You close this, Cindy. Go ahead.
I just want to pray for those whose eyes don't see and whose ears can't hear. They can't see your amazing awesomeness, Lord. Please, we ask that you would send your Holy Spirit. We know that it's not your will that any should perish, but that all would come to repentance, Lord. So that's what we ask for, and use us to that end. In Jesus' name, amen.
Amen.
Majesty.